Hello, everyone, and welcome to today's webinar, Cosmic Combining Expert Curation with Scientific Innovation in a Gold Standard Database for Precision Oncology. I am Marie Stone of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by QIAGEN. To learn more, visit QIAGEN.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. I'd like to now welcome our speakers. Dr. Zbyslaw Sondka, Senior Scientist, Cosmic, Catalog of Somatic Mutations in Cancer, Welcome Sanger, and Rebecca White, Scientific Communications Manager, Cosmic, Catalog of Somatic Mutations in Cancer, Welcome Sanger. Dr. Sondka, Rebecca, you may now begin your presentation.
Thanks so much. So I'm going to kick off with just a bit of a description of why Cosmic, why Cosmic exists, what we do, and how we do it. And then I'm going to hand over to Zbyslaw to talk about a bit more about our products and how you can get the most value from them. So starting with the why. We exist to accelerate the advancement of cancer research, be that diagnosis, treatment, or prevention. And ultimately, we do that to improve the length and quality of life for all cancer patients. Our vision is that one day a patient will go into a doctor's surgery, and as soon as they hear a doctor say to them, "I'm sorry, you have cancer," it's followed up immediately by the words, "But it's okay, we know exactly what to do about this and exactly what your treatment journey will be." What do we do within that context? Well, we collate, streamline, and quality filter global cancer somatic mutation data. We make that data useful and easy to understand, and we draw out salient insights. The idea is that our database will accelerate research without compromising on quality. And exactly how we do that is through expert curation, data interpretation, and visualization. We're experts in identifying data, extracting it, standardizing it, and presenting it. Now, I'll go into a bit more detail on this later, so don't worry about the notes here. And I just wanted to kind of start with that why and positioning ourselves within cancer research. So as many of you will know, cancers are no longer thought of as one single disease. It can be thought of as a group of diseases that share common characteristics that give them an advantage and abnormal behaviors that help them to grow and thrive. And these are really nicely described through the hallmarks of cancer. Excitingly, the hallmarks also represent really good strategies for tackling cancers. If you can take away things like their ability to grow vasculature and their food sources, if you could get immune cells to recognize the cancers, or if you could somehow stop the cancers from growing and spreading, that would be a really good way of stopping things. But it's a bit more complicated. We know that obviously there's not one single mechanism by which tumours and cancers achieve these things, and they do it in multiple different ways. And sometimes, even if you tackle one aspect, there might be a redundant mechanism where cancers are doing it in multiple ways. And I'll come into more on this later and why it's important in the context of somatic mutations. And of course, we also need to think about cancer on lots of different levels of biological organisation, in terms of the environment and what sort of mutagens we're exposed to the body and how all of our bodies are slightly different, through to the actual environment, microenvironment, and specific tissue differences, to the cellular level and what's going on there, to the proteins and their dysfunction and dysregulation, and finally down to somatic mutations or mutations in the genes. And all of these changes participate in the development of the disease, but it starts with our genes. And that's why somatic mutations are so important. They help us to understand the bigger picture. Through analysis of somatic mutations, we're able to link DNA change to protein dysfunction. We're then able to associate those known functions of the protein with biological processes and identifying the pathways and processes involved in disease progression. Again, this is important because if we think back to the fact that you can get resistance or certain treatments in isolation don't work, that's because a few mutations, if you look at them, might be impacting multiple pathways, which give the cancer the same ability to maybe grow uncontrollably. So you need to think about how you can target multiple pathways, and then choose an optimal therapy regime or propose a new therapy regime, or in fact, tell a patient we know this treatment won't work, saving months of unnecessary treatments. And finally, it's unpicking the etiology of cancer. If you can combine mutations with information about things like environment, you can begin to understand what's causing cancers for individuals. Now, so in the ideal world, what we'd have is genetic information linked to the cancer information, linked to treatment information. But again, another challenge is there's just too much data and there's not enough time. In 2020 alone, there were more than 30,000 publications that linked cancer and mutations. And that's just way too much for any single person to keep up to date with. And they're still missing information. We don't know exactly which mutations are driving cancer, the functions of the products of those mutations, or which mutations could be good candidates for drug development. We know some of it, but not all of it. And we also end up working in niches and focusing on, say, a specific cancer type or a specific protein type. So what you need is a big database that collates all of this so you can begin to see the patterns across cancer types, across mutation types, and how maybe one treatment may be applicable in lots of different types of these diseases. So as I said, that data becomes useful once collated, standardized, and structured so that insights can be extracted. And that's exactly what Cosmic does and what we're experts at. And in fact, we've been experts for 20 years. We started out in 2002 at the Sanger Institute. That's before the Human Genome Project was even completed. It's a spreadsheet of the Sanger Institute's Cancer Genome Project to keep track of genetic mutations. And it started out as just 4 genes: BRAF, HRAS, KRAS, and NRAS, just 67,000 samples and 10,000 mutations. It was so useful, it's been grown and it's sustained. To today, we include all genes, more than 1.5 million samples, and 23.4 million mutations are recorded. Our data resolution is unparalleled, and our experience is unparalleled, and that's why we are the gold standard database. So how exactly do we go about doing this? Well, this is kind of a broad summary. We gather data from high quality and trusted resources worldwide. Mainly, this is scientific literature, but we also get data from some of the wider cancer genome consortiums and data from some smaller studies. And more recently, we've also been getting data from clinicaltrials.gov and other clinical trial resources in our actionability product. We combine all this data and we run it through a large standardization and quality control pipeline and map it to other existing resources and use those to annotate our data, such as Ensemble. And then that is then produced into a range of products. The most common one is our download file, but then we also have a suite of data visualization and data interpretation tools, which will be gone into in more detail later. But I'm going to spend a bit more time focusing on where our data comes from. So we have two different types. We have this broad genomic data, and that's, as I said, comes from these from semi-automation annotation of the big genome screens, and also genome-wide studies that are published in the scientific literature. We take the supplementary data sheets and run them through genetic annotation systems. And then all of that is mapped to both Human Genome Assembly 37 and 38, and you can toggle between both on our website or download data from both. But our real kind of area of expertise is in manual curation. I said earlier that we were first formed in 2002, and we still have some of that curation expertise from when we were first formed in the team and being passed down. Our curators are postdoc level experts who they have brilliant expertise in being able to extract data and high quality data from publications. And it's not just the gene data, they also extract data on things like cancer type and stage, and also some kind of wider clinical metadata around sex, age, family history, prior treatments, certain environmental factors, as well as the mutation status and sample source. And this deep genetic data isn't random. It actually comes from our Cancer Gene Census. So the Cancer Gene Census is currently 729 genes which are known to have to be drivers of cancer. And that's the literature proves that they have a mechanistic role in driving cancer forward. There's tier 1 and tier 2 genes. Again, this will be discussed in a little bit. But those genes are where we focus our gene curation because we know they are the relevant genes and it removes some of the noise of irrelevant things. But we also have disease-specific curation. So each release, our curators will focus on a specific disease, and usually this is rare cancers. Last release, it was metaplastic breast cancer and other cancers of the female genital tract. And this release, it will be head and neck cancer. Now, what's interesting about the disease curation is through reading all those papers, our curators identify new genes that should be put forward as candidates for the Cancer Gene Census because they're the role in cancer they're seeing in these papers. And then that gets handed over to our Cancer Gene Census expert who will then validate or invalidate and then add that gene either as tier 1 or tier 2 to our census. So in our upcoming release in V96 in May 2022, we have a focus on head and neck cancers, which account for 3 and 5% of the US and European cancer incidence. Rather than focusing broadly on all head and neck cancers, they've done a specific focus on the rarer forms of head and neck cancer, those that affect salivary glands, sinuses, or muscle and bone in the head and neck. That data has come from 56 publications. And this is a really nice example where PRKD1, a serine threonine kinase, was added to the Cancer Gene Census as a tier 2 gene because it was found to have a role in cribriform adenocarcinomas. So this is just to show how all of our data links together. And then the end outcome in terms of pure stats is what you're seeing here. We have a total of currently 23 million mutations from 1.5 million samples, 28,000 papers, and 39,000 WGS samples. And we also record things like copy number variants, fusions, differentially methylated CPGs, and gene expression variants. So now I'm going to hand over for a more detailed look at our products and how you can use them.
Thank you very much, Rebecca. So that was an overview of the whole database. Now I will tell you a little bit about how this data can be used in real life. You are all aware that there are many different types of cancer. One of the most common cancers is the lung cancer, and 85% of all lung cancer are usually non-small cell lung cancers. They are rarely diagnosed before they become metastatic, and they are major cause of cancer mortality. So, what cosmic data can tell us about lung cancer? What genes and what dysfunction are most typical for this subtype of cancer? From this list, you can see that 3 most frequently mutated genes in this type of cancer are EGFR, KRAS, and TP53. So that's the first information. We have these 3 genes that are potentially causing cancer and potentially can be targeted. If you go deeper into the data, we of course can see much more. KRAS. This is a known oncogene, and on this slide you can see the distribution of mutations in cosmic in lung cancer patients across this gene, and you can immediately see that practically all mutations are focused around this one spot, which is glycine 12, and this is typical pattern for an oncogene which suggests function. This is typical for oncogenes because it's much more hard to bring to cause the gene to be more active than to just destroy its function. So in this case, very precise change is required, and this is what we can see in cancer populations. Going deeper into that, you can see what is the composition of these mutations that affect mostly these 2 amino acids, 12 and 13. So you can see there are different consequences, and you can see that even within the same codon, not all nucleotides are equally affected. So one of the reasons of that situation is the limitation of the genetic code. So only certain changes bring selective, bring this desired change that actually drives cancer. And the other reason is what factors are actually responsible for mutations. And here you can see a comparison between lung cancer and colorectal adenocarcinoma. On amino acid level, it's exactly the same change. But you can see that on the DNA level, most frequent mutations are slightly different, and this is caused by the different exposure to environmental factors. In lung cancer, the major factor is tobacco smoke, which results in mostly G2T changes, while in colorectal adenocarcinoma, these may come, for example, from heterocyclic amines, and the result is mostly G2A. Still, on the amino acid level and on the protein level, the change is exactly the same, and the same consequence, increasing proliferation of cancer cells. Another gene has slightly different function, P53. This is a classic tumor suppressor gene. You can see that the pattern of mutations in that gene is slightly different. They are spread more evenly across the whole gene body. And especially at the bottom track, insertions and deletions, you can see lots of mutations are there, and many of them results with frame shift or truncating. Anywhere the mutations happen, they just break totally the function of tumor suppressor gene, which cannot protect the cell from oncogenic transformation. And these are, again, these mutations that result in nonsense substitutions or frame shift insertions before it ends, and it's not active in the cell. Then third of the gene GFR is again, as you can see here, very nicely focused peaks of missense mutations, gain of function. But in this case, they are not clustered just in one place, like in case of KRAS, but we have several categories of gain of function mutations. And knowing about these mutations is quite important because they are actually used to design specific drugs that act against mutated forms of this protein, and are more or less selective. So an example is mutation T790M, which has a drug designed to act against it, osimertinib. Then we have another cluster of mutations in that gene, which are in this case not simple substitutions, but insertions and deletions. But what's different between these mutations and mutations we have seen in TP53 is that these are in-frame in those. So that means that the sequence of the protein after the mutation is changed, and only small fragment of the gene is either added or removed without changing the main function of the gene, but with changing the control mechanisms that are normally protecting protein from being hyperactive. I mentioned T790M gatekeeper mutation. This is quite important mutation because it makes lung cancer resistant to some of the precision oncology drugs, and they are often arising as a result of cavity therapy. Because it occurs predominantly under the therapeutical regime, and after this drug is removed from the body, then these mutations tend to disappear. So this creates an opportunity to precisely design the therapeutical regime to control occurrence of these mutations. But you need to know that this mutation is occurring or not, and to be in control of that process. Okay, excuse me, I just got a message that my sound is not very well. I'll try to disappear to improve the quality. So that was the lung cancer, but of course there are much more types of cancer in Cosmic, and you can see similar data for all of them. And I would like to give an example how you can compare different cancers. And in this case, I'll start with hematologic neoplasms. You have three major subtypes here: acute myeloid leukemia, myelodysplastic syndrome, and myeloproliferative neoplasms. And these are very general subtypes of blood cancers, and you can see that already on that level the gene sets are slightly different, but of course you can go deeper. So that gives you this high resolution of phenotypes. You can check the set of genes which is specific for certain cancer type. So if we go deeper and compare myeloproliferative neoplasms, we can see different rare cancer types, and I would like to focus especially on mast cell neoplasm. This is very rare cancer. Most cancerologists see them only once or twice in their careers. And the interesting thing about this type of cancer is that the relevant driver of this disease is KIT, which is an oncogene. And this oncogene is known from non-blood cancer, which is in this case gastrointestinal stromal tumor. So exactly the same gene is responsible for development of two different cancer types. But when you have a look at the mutational profiles within these two diseases, you can see the differences. In case of mast cell neoplasm, it's mostly phosphotransferase domain that is affected. In case of gastrointestinal stromal tumor, these are mostly mutations that affect juxtamembrane domain. And that shows you again how different tissues development of different cancer types. Even though it's the same gene, not always the change that causes this gene to drive cancer is the same. And this is very important to know, again, to design diagnostic tests to interpret variants of unknown significance and to identify pharma targets for precision oncology. So this is about somatic mutations, simple somatic mutations, but of course there are other changes to the DNA that can drive cancer, and gene fusions are a famous example of that. Cosmic has a resource that currently describes 305 gene pairs, and this is again manually curated, so the quality is priority here, and you can explore gene fusions as well. So just to summarize this, with all this data, we can identify typical for each cancer type and with single type. Then, to certain degree, just by looking at the patterns of somatic mutations, we can predict their impact. Is it gain of function or loss of function? Are we talking about tumor suppressor gene or oncogene? And then we can find some clues about the origin of mutations. Caused by tobacco smoke, or is it caused by... But contrary to what it says here, they actually don't answer thousands of genes and millions of mutations drive cancer and why. To some degree, it does, but typically, when we have a cancer sample, we have thousands of mutations there. So looking for these very few mutations that are actually matter there is quite complicated. Then it doesn't answer the question which mutations create therapeutic opportunity and which make the therapy ineffective. Then another interesting question is how we can better determine what mechanisms drive cancer and, for example, genomic instability. And Cosmic has several products that are built on top of the main database. Rebecca already mentioned the oldest Cancer Gene Census. And Cancer Gene Census is manually created resource that gathers genes for which we have very good evidence that they are involved in cancer driving processes. And to add to that, we not only say genes are driving cancer, but we also say how. And this is another resource within Cancer Gene Census, Hallmarks of Cancer. So again, Hallmarks of Cancer tell you which biological processes are disturbed in cancer cells. So for example, is it DNA repair that is damaged? Is it proliferation? Is it apoptosis? And each gene is described in terms of hallmarks of cancer, and manually curated information tells you very briefly what hallmarks this gene is involved in. So for example, TP53 is involved in control of apoptosis. You can find it on these pages for every gene, and you can find links to the source of this information. You can go straight to PubMed. So that was Cancer Gene Census. Sorry, I didn't change the slide. And of course, it's not only about mutations. So another thing is that all we're talking about were mutations. They describe dysfunction, but many of these genes are also changed through differential expression. And Cosmic has these differential expression variants too. And you can see on this example that APC, which is non-tumor suppressor gene, very important in colorectal cancer, where it is usually affected by inactivating mutations. In ovarian cancer, is mostly downregulated on the expression level. So for the same gene and different cancers, you can see also different ways this gene is affected. And then one of the questions was, if you identify the right mutations, what can you do about this mutation to help patients? And to address that question, we have created another manually curated resource, Cosmic Actionability, where for each mutation and cancer type, we are curating all the drugs that can be used or that are during clinical trials at the moment. So basically, it covers the current state of precision oncology. And to help navigating through this resource, we introduced different actionability ranks. With first one, drugs are already on the market and approved, but also we present drugs which are in late phases of clinical trials and met primary objectives. Also, drugs in trials without primary objectives reached, and we add some case studies. This resource is updated 4 times a year, so all the data that we provide there are quite up to date. As for the sources for actionability, we use clinical Excel database records, but around half of the data are actually coming from additional data sources, like literature. So many of the clinical trial results are not being updated in the clinical trials database, but instead the results are published in the literatures. We look for that and we integrate data source to give you the most comprehensive information. Also, if the trial terminated, we aim to because sometimes it's not just lack of efficacy of the drug, it can be business reason. And in such case, we believe it's good for people to know that this has happened and this drug actually still can be potentially good. So this is the summary. At the moment, we have almost 50 genes fully curated. Over 200 genes are mentioned there. This resource is under constant development, and as I said, priority for us is to keep it up to date at all times. Then, of course, I already mentioned certain mutations result in resistance to certain therapies, and Cosmic does gather information about such mutations. So mutations that bring resistance to particular drugs, which mutations occurrence should be monitored, and then the therapy can be changed. Cancer Mutation Census, another product, tells you which of the thousands mutations actually matter. In this example, you can see NRAS, I was talking about before, and Cancer Mutation Census uses different biochemical parameters and properties, manual curated information, and with certain algorithms like DNDS, for example, that measures the selective pressure that acts on certain amino acids in cancer samples. And by integrating all these, this is mostly again G12, G13, and N61, and all this information is clearly presented. So we propose our own ranking of the mutations, but rule-based, and you have all the components of the data that we use to create that ranking, scrutinize for yourself, and to work out your own ranking if you are not happy with ours. On the mutation level, every of the 5 million subcoding mutations in Cosmic is described with all these values, and we have, of course, statistics in Cosmic across all the diseases, so you can see what frequency this variant has in every disease. You can see clean var information if anything has been manually curated about this variant. It's known as cancer driving. So we focus only on cancer-specific annotations from CleanVar. On the other hand, variants that are to be frequently seen in normal populations as germline variants are not likely to drive any cancers. We use NOMAD database to provide frequency across all populations annotated. Check every variant against this information here, plus DNDS. If certain variant has positive DNDS score in certain cancer, then we report it here. TP53 that I mentioned before as a classic tumor suppressor gene is because you can see three spikes of very recurrent mutations, which, as I said, are typical for oncogenes. And that is a very interesting case because TP53 is known as a handbook example of tumor suppressor gene. So what these three peaks do? Cosmic 3D, another tool that we have, is helping answering this question, because even though you have three distinct peaks on the gene length, when you project these mutations on the 3D structure of the protein, you can see that with this red color, they all really cluster in the 3D space around the sites that bind that TP53 uses to bind to DNA. And they change the affinity of TP53 to DNA, and they are, in fact, gain-of-function mutations. Give TP53 slightly different function, and instead of preventing cancer, it actually, in certain cases, drives it. Another example is EGFR, and another feature of Cosmic 3D. We can see the difference between wild type and mutated amino acid. And here, with the orange, you can see the mutant protein, and with the pink, you can see the wild type protein. And you can see that better on that figure, that the orange protein, the orange space actually invades the space occupied by the small molecule drug, and that shows the mechanism of drug resistance in EGFR. Then the slide I was mentioning before, the difference between exposure to different mutagens. We can see the result of that on the DNA level, but is there anything more we can tell about it? And in fact, there is. Cosmic also presents mutational signatures. This is a very useful thing because you don't need to do any additional experiments. Basically, you take the next-gen sequencing result for a patient sample, and you analyze single nucleotide substitutions with their surroundings, and then you see which types of alterations have what frequency. You end up with a histogram, you can see on the right here, that shows you that in this sample you have quite a lot of C2G changes and C2T. That's the first stage. This is your mutagram. Then we can use some deconvolution algorithms to identify patterns typical for different mutagenic factors. So the mutational profile then is deconvoluted into the mutational signatures that allow you for identification of mutagens that were causing the mutational process within the sample. Cosmic has a catalog signature and explore, and then you can compare signatures in your samples with signatures in Cosmic catalog. This is still ongoing and growing, and we are adding new signatures, but they are again manually curated, and only when they are accepted by the scientists working on it, we add it to our catalog. So it's quite conservative, and if you use these signatures, they are something you can compare with other people. Particular application of the signature concept. There is a signature that is typical for the damage of homologous recombination repair. And in this case, you can of course see mutation in BRCA1. This is similar change. In this case, it's not about looking for mutations in genes involved in that. This is measuring the effect. So it doesn't matter if the damage of the homologous occurs through mutation or through changes in expression. The genes can be applied. So all these data are available for download for academic users directly from Cosmic and for commercial users through QIAGEN. I'm happy to answer all your questions, and I'm sure so is Rebecca. Thank you very much.
Thank you, Dr. Sondka, Rebecca, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question on the far left of your screen. We'll answer as many of your questions as we have time for. Let's get started. Our first question is, how do you decide which papers to curate?
I can take this one. So I'd love to say there was a very specific formula to it, but really it comes down to the expertise of our curators. Obviously, as I said, each of them are responsible for certain genes, and they have searches in place, which means that they're constantly downloading papers that are related to those genes, and they'll use a bit of their own instinct and insight. So if it's a very common gene and very common mutations are being reported in a paper, maybe they will put that to one side and focus on something novel that's new, that adds some new insight to our database. That's not to say they wouldn't curate that paper, but it's that kind of instinct and insight around quality. More specifically, there are some minimum requirements in place for a paper to be included. There's details about this on our web pages under the submissions page, but it's things like the minimum requirement for a sample to be included. It needs to have full mutation details provided at either nucleotide or protein level. And the same if there's samples with more than one mutation, we need to know which specific mutations occur together in any given sample. Another interesting thing we do is we do accept author submissions, and we won't guarantee that we will curate it if it's submitted. It does need to meet criteria, and it may take a while, but that is also an aspect that we have in Cosmic, which can also be found on those submission pages.
Thank you. Next, how does Cosmic help users get maximum value from its data?
I'll take that one. The data is the main database. As you saw, we have worked on multiple additional products that help interpreting these data. So it's not only about what is changed in cancer, but also why and what are the consequences and what we can do about it. And we also have help pages. I think, by the way, that's the one of the next questions, showing how to present data. Another thing that we are currently preparing are scripts to analyze the data in Cosmic, and they are standard. They will be standard protocols for getting the answers that users want to have answered. And if you can suggest any problems that you are dealing with, what would you like to see, then we will be very grateful for your feedback on that, and we can prepare proper software that analyzes the Cosmic data.
Great, thank you. Next question, where can I learn how to obtain cosmic outputs that you are presenting here? Is there a training course?
Yeah, so I think we just covered that a little bit, but I just wanted to cover, with part of our science communication strategy, we're exploring how we can be running some more events, tutorials, to really add value and to help people make the most of our data. At the moment, we've not got any of that strategy in place. We're trying to understand what the biggest needs are. But if you follow us on Twitter, and also if you sign up for our mailing list, which you can do when you sign up to Cosmic, we will be putting further details out and asking some more questions around that. In addition, we do have tutorials. So for instance, with Cosmic 3D, which you just saw being presented, we're going to be doing a launch of a revitalized version, and that will come with a tutorial video, and we'll also be running an event to talk you through Cosmic 3D in more detail to try and understand how to make the most of that data. And we're hoping to do that more product by product as we go forward. So it's not quite there yet, but it is in the pipeline, and we are hoping to produce more like this. And as you just heard, the more you tell us about what you need and what you're looking for, the more we can then begin designing those resources.
Great, thank you. Next question, what is the breakdown of adult cancer versus pediatric cancer representation in Cosmic?
This is something you can very easily extract from the download files. I'm not quite sure if the website can do that, but as Rebecca mentioned, this is one of the data points that our curator gathers. So whenever possible, we either have a distinction between child and adult, depending how detailed the information in the paper is. But where the accurate age is given in the paper, we also have that information. And as I said, it is contained in the download files. If you have any around that, and we'll be happy to help you with that.
Great. Well, thank you again, Dr. Sondka, Rebecca, for your time today and your important research. We would also like to thank LabRoots and our sponsor, QIAGEN, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today, and those submitted during the on-demand period, will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.